Low back pain is the leading cause of disability in the world, and this is often linked to the intervertebral disc. The cells in the disc respond to mechanical loading, and this interaction between mechanical loading, cellular behavior, and the tissue structure can lead to a vicious cycle of degeneration. While intervertebral discs have previously been cultured under simplified loading conditions to study these interactions, the 6-axis bioreactor at the University of Exeter has been developed to complete dynamic 6-axis intervertebral disc culture tests. This provides a way to investigate the response of the disc tissue and cells to the complex loads and movements that occur during different daily activities. We developed the activity profiles to apply to discs by combining the Harmonised European Time Use Survey, which documents the types of activities that people do and how long they spend doing them, with the OrthoLoad database, which has 6-axis load data for patients with instrumented vertebral body replacements completing different activities. This allows us to create different 24-hour activity profiles based on real populations and includes the replication of sleeping and more sedentary activities, as well as different light and moderate intensity activities such as walking, cycling and lifting weights. Our initial research has focused on how a 6-axis activity profile affects the disc compared to similar loading in just axial compression and no loading at all. During these culture tests, we were able to see that the stiffness of the discs and the recovery during periods replicating sleep were consistent across the 7 days of testing. However, at the end of the tests, we found that 6-axis loading led to a lower percentage of live cells compared to the unloaded discs or those loaded only in axial compression. This doesn't mean that more movement is bad, but it does suggest that the more realistic 6-axis activity profile creates a more challenging environment for the disc cells. These results are important when considering new treatments such as regenerative therapies, as they will need to manage the increased demands of 6-axis loading if they are able to be successfully used in the human body. These tests are just a starting point, and the unique 6-axis bioreactor at the University of Exeter provides the potential to complete further studies, including tests that use different activity profiles and longer culture periods, to understand more about the biomechanics of the spine, complete preclinical testing of medical devices and therapies, and investigate the interaction between the mechanical and biological environments of the intervertebral disc in ways that have not previously been possible. This research was completed in the Department of Engineering at the University of Exeter, with support from researchers at the University of Queensland, and it was funded by the Engineering and Physical Sciences Research Council.